The advantages of fluorescence glucose biosensors includes the greater sensitivity to low glucose concentration. This means that even if the sample such as blood have low concentration of glucose, this device is still capable of detecting it. Next, it is accurate in hypoglycemia range. Hypoglycemia range is low blood sugar, and this action is done by binding the proteins with dissociations constant in range. Next, there is less need to recalibrate in response to local tissue reaction around the sensor. And with this device, we no need to implant transmitter or power source for wireless communication of glucose data, so it is more convenient for the user. However, there are some major disadvantages of fluorescence glucose biosensor compared with the commercial glucose biosensor. Firstly, it is very sensitive to local pH and oxygen. This will eventually affect dye response. Next, it creates damaging foreign body response. Next, the implanted dyes can be potentially toxic, especially if the implanted fluorophores can't be fully removed from the patients. Next, we are necessary to always carry a light source for implantations of this sensor. This type of glucose biosensor is considerably high cost because we need the usage of fluorophore, light source and dye. We move to amperometric glucose biosensor. The advantages include high selectivity and specificity due to the enzymatic reaction. Next, it is relatively simple because they use electrochemical detection method. Moreover, there will be potential for wide applications because it is not only restricted in testing the glucose in human blood, it is also can be applied in food and beverage industry in glucose detection. Furthermore, this type of glucose biosensor is highly sensitive because of large current density that enable electrode miniaturization. That is, it can be manufactured into a smaller size device. On the other hand, there are some cons of amperometric glucose biosensor. Firstly, it is highly dependent on temperature and pH. Since this is a type of enzyme-based detection, so it is essential to create an optimum temperature and pH. Next, it is easily interfered by chemical presence in sample matrix. The chemicals can include small metabolites, cells, and macromolecules. Another disadvantage is that electrode pre-treatment is always needed for obtaining a reproducible surface. Lastly, permanent data correction is always needed due to the effect of background solution. This is because of the interference with non-specific particles that is present in the sample solution.
Greetings to Dr. Tan. I'm Gong Tin Ho, and I'm going to talk about past studies on glucose biosensor. Okay, actually, the determination of the glucose level is significant in biological and clinical, fermentation and food processing, as well as chemical samples. Therefore, many studies has been done since the introduction of glucose biosensor. The development of fabrication of a cost-effective, simple, accurate, portable, and rapid sensor for glucose biosensor are socially crucial for us. Okay, I'm going to talk about polyaniline first. This is a material that many research has been used for enzyme metrics for bioenzymatic glucose biosensor. Polyaniline has got recent attention because of its interesting characteristics such as high conductivity, optoelectronic properties, environmental and electrochemical stability, biocompatibility, low cost, and easy doping and dedoping chemistry. Okay, as you can see, this polyaniline plays significant role in glucose sensing system. It provides a favorable matrix for the immobilization of glucose oxidase enzyme. It also acts as electron mediator and facilitates the charge transfer in enzymatic redox reaction. This is a summarized table on the past studies using polyaniline as the matrix. As you can see from this table, all enzymes using are the glucose oxidase enzyme, and of course polyaniline as the matrix, and sometimes they also include other materials for their experiment. For the enzyme immobilization technique, they also use different like entrapment, covalent, cross-linking, and adsorption method. For different kind of this method, we can see that there is different in selectivity, detection range, and the detection. I also read about a few articles on these past studies on glucose biosensor. As you can see, all these dark boxes are actually the glucose source for the glucose concentration determination. All these articles, the researchers are using different kind of methods, materials, and conditions to conduct their experiment. In this slide, I will only talk about what type of glucose biosensor they use for their experiment. For grape juice, honey, and glucose purchase, they are using this GOD base, which is a bioenzymatic glucose biosensor. This glucose biosensor has the same principle as amperometric glucose biosensor, which they observe the hydrogen peroxide release to calculate the glucose concentration. Then for this bioreactor, they are using this FIA base, which is a flow injection analysis glucose biosensor. This biosensor can continuously monitoring the glucose concentration in the bioreactor tank. Then for human blood plasma, they are using this self-powered glucose biosensor. Actually, this glucose biosensor are using the biofuel cell as the power source. So we don't need any other external power from the outside to operate this glucose biosensor. Okay, so I will continue to talk about the articles I read. For this tear sample, they are using this this paper-based glucose biosensor. As you can see here, the tear sample is just put on top of the surface with these reagents, different kind of reagents. Then we can, after the detection, we can read the glucose concentration from a device or computer. From this article, they even said that this device provide analytical reliability and no statistical difference when compared to the data using a commercial glucometer. Then for this smart contact lens, they are using this fluorescent-based glucose biosensor. This contact lens, the aim is to continuously monitoring and detect the glucose level in a person. Then lastly, for this conjugate protein stop, they are using this FRET-based glucose biosensor, which is a fluorescence resonance energy transfer glucose biosensor. Even in protein, there are many glucose inside, so they conduct this experiment. That's all in this slide. Thank you.
So I'm going to explain about application of glucose biosensor. So application, before this, we just use a simple application of glucose biosensor. We just use a pinch our finger with a nail, nail something. Then we can get our blood, so we can show that our blood glucose is what are the type and what high is it. So the new application of glucose biosensor is saliva glucose biosensor, paper patch, and graphene patch. Saliva glucose biosensor is what we use, and we take a paper and we put in our mouth, a paper that already have been modified, so we can put it in our mouth. So it will we will control it in our phone, and we can get the result of our glucose blood pressure, all of that. So next is have a pepper patch. This is pepper patch. Pepper patch is like plasticines we put in our arms or our legs or something like that. So we can use monitor in the computer, so we can know that our glucose percentage in our body. So next is a graphene patch. Graphene patch is a new one. It's the newest because we will show it on the paper. If we put in our arms, we can see that our glucose level and our glucose things in this paper patch. So these are the new 3 things in the paper patch, in the glucose Biosensor.